Hi class, my name is Alicia Yergi. Today's date is July 19th, 2021. The time is 7:25 p.m. This is week 2, lab 3, dilutions and plate count experiment video. So what we did first, of course, we cleaned off the area with the bleach solution and laid our chucks down just in case there's any spills. Put on our PPE and then got everything ready for our experiment. So I have 6 test tubes here, 6 agar plates here. Each one is labeled 10-1 all the way to 10-6, so they correspond with each other. So 10-1, 10-1, and so forth. So what we're going to do is we're actually going to put the yeast contents into each of these tubes. I already pre-filled the tubes with the distilled water. Of course, my pipette was sitting in the alcohol for at least 20 seconds to sterilize. And then after I shook the pipette clean of alcohol, I went ahead and filled my 6 test tubes with 2.25 mLs of the distilled water. Yeast has been setting already for 10 minutes. And so what I'm gonna do is I'm actually gonna take the pipette and pull up 0.25 mLs of the yeast and place it in the test tube 10-1, and then I'll just kind of pipe up and down the solution in 10-1 like this. So my pipette's been sitting in the distilled water after I had pre-filled everything. So I'm gonna go ahead and get 0.25 of the yeast content. I'm gonna grab this right here, test tube. Okay, and we're gonna add that in here. Make sure all the solution is out. So I'm just gonna kind of almost like rinse the pipette in the solution. Make sure everything's out. And then I'm gonna go ahead and repeat the steps for all the test tubes. I had already started placing the contents. I did a practice run before video recording. So 10-1 has already been done. So what I did was I took some solution from 10-1 and played it on the agar. So approximately 4 drops of that solution after everything was diluted. 4 drops here, opened up the agar plate. After dropping, put the lid back on, swirled it to make sure the solution covered the agar. And I'm gonna do the same steps for each of the agar plates. I'm just gonna finish the dilution. So I'm gonna take, again, 0.25 of the solution in 10-1, and I'm gonna transfer it to 10-2. Okay, 10-2. And again, we're gonna kind of pipe up that solution up and down. I'm gonna place this one back down. I'm gonna take this one, and now I'm going to pull up another 0.25 and transfer it to the 10-3 tube. I'm going to put the 10-2 back down, take the 10-3. Same thing, I'm going to take 0.25 And I'm gonna go ahead and place it into 10-4, same thing. And make sure that all the contents are out. Then I'm gonna take this and transfer into 10-5. Okay, last transfer to 10-6, and then after that, I'm gonna completely rinse this out in the distilled water. And then I'm gonna take from 10-2 because I already did 10-1. Just make sure that's all rinsed out. And then I'm gonna take my 10-2 plate and the solution, and I'm gonna take approximately, or just drop some of the content and add 4 drops to the plate. 1, 2, 3, and 4. Okay. And then I'm gonna cover that up and kind of swirl the plate around to make sure the fluid covers the agar. And then we're gonna go ahead and do the same for the remaining plates, 10-3, 10-4, 10-5, 10-6. Let it sit for 48 hours and check on the growth, and then we will post the results video. Thank you.